Hi, my name is Sriyanka Lahiri and I work at the University of Florida's Gulf Coast Research and Education Center. Today's talk focuses on the common insect and mite pests of strawberries in Florida and their management. Thrips are proving to be most difficult to control currently. So I will start this discussion with thrips and focus more on thrips management. In Florida, two species of thrips cause economic damage. They are chili thrips and western flower thrips. Chili thrips start infesting strawberries right after being transplanted. They feed on the young foliage, flowers, and fruits. Chili thrips is an invasive species from Southeast Asia. The western flower thrips feed on the flowers and fruits of strawberries only. In the picture on the right, an adult chili thrips and several larvae are shown feeding along the veins of the leaf. The picture on the left shows feeding damage on young expanded foliage from chili thrips. This is the size of strawberry trifoliate that you should sample in the field for chili thrips monitoring. Western flower thrips occur on flowers and fruits, therefore sample flowers and fruits to monitor western flower thrips. You can also use yellow sticky cards to monitor chili thrips and western flower thrips. As soon as you see darkening of leaflet midrib, starting at the base of the leaflets, it's time to make a management decision. High chili thrips infestation leads to leaf necrosis and death. Thrips infested fruits turn bronzed and cracked, and the thrips larvae, be it of chili thrips or western flower thrips, will remain well hidden underneath the calyx of the fruit. A recent field study was conducted by my student in 4 grower fields in the Plant City area. Young trifoliates were sampled on a weekly basis for chili thrips larvae and adults. The results from 3 fields are presented in the graph. The y-axis shows mean larval count per 5 trifoliates, and x-axis shows the sampling dates. The number of larvae show 2 peaks at all 3 fields around the same time. Also, field D showed the earliest detectable larval counts experienced higher population through the rest of the season when compared to other fields. Efficacy of conventional insecticides to suppress pest thrips were also tested in the field during 2019-2020. The treatment legend shows trade names on the right and modes of action on the left. Figures depict chili thrips and western flower thrips adults or larvae on the y-axis and treatments on the x-axis. Radiant was most effective in significantly reducing chili thrips adults and larval stages. Exirel at the high application rate of 20.5 ounce per acre and Minectopro at 10 ounces per acre was comparable to Radiant for chili thrips adult suppression. Minectopro, Exirel, and Radiant significantly reduced western flower thrips larvae but did not make any suppression on the western flower thrips adults. A greenhouse study was also conducted to test the efficacy of a predatory mite Amblyseius swirskyii in suppressing chili thrips when compared to a biopesticide Captiva Prime and the conventional pesticide Radiant. Potted plants of Florida Brilliance cultivar was used for this experiment. Biopesticides was as effective as Radiant, which is spinatorium, in suppressing the adult chili thrips populations. The predator, Amblyseius swirskyii, was as effective as the biopesticide in suppressing the adult thrips population, but not as effective as Radiant. The larval stages of chili thrips were controlled equally well by both Radiant and the predatory mite. However, the same could not be concluded for the biopesticide Captiva Prime. Most thrip species that occur in fields are harmless, and some are even predatory in nature. Only a couple of species, such as the chili thrips or western flower thrips, cause severe economic damage to crops. Therefore, monitoring the fields and correct species identification is a very important first step to avoid secondary pest outbreaks or resistance development in pests. Rotate modes of action when relying on insecticide application and select softer products to conserve natural enemies. You can also consider releasing predatory mites, at least in high thrips population areas of your field. Keep in mind that predatory mites are good at suppressing thrips larvae, but not the adults. Conversely, most insecticides are effective in suppressing adult thrips, but not the larval stages. When using predatory mite releases, wait at least 7 days to release predators after insecticide application. I have highlighted some of the insecticides that were tested for efficacy at the research station. Please pay attention to the modes of action for each product mentioned within the parenthesis. The website link will take you to the entire list of the IRAC modes of action. Do not apply similar modes of action back to back. You may also consider mixing an insect growth regulator to suppress the larvae. Remember, label is the law. Therefore, check product label for making any tank mixing. Mites form another pest complex in strawberries, especially the two-spotted spider mites, seen here in the picture on the right, with eggs that are spherical in shape and laid on fine webs on the leaf surface. Initial infestation becomes evident as stippling on mid-tier leaves. Severe infestation is clearly visible because of webbing on the plant. Cyclamen mites are also a pest of concern since they hitchhike on transplants from nurseries. They feed on young folded trifoliates, leading to stunted leaflets in the center, also called the compact leaf mass formation. Adults, nymphs, and eggs can be found underneath the fruit calyx. Damaged fruits turn bronzed and crack, just like from thrips feeding. Adult mite does not have any spots and appears clear and light pinkish in color. The eggs are oval in shape and laid at the edge of the leaf hair as shown in the picture on the right. The best way to manage the two-spotted spider mites in the field is augmentative releases of the predatory mite Phytoceus persimilis. This is called biological control. When selecting miticides, avoid broad-spectrum products. Certain miticides, such as Portal, is effective in controlling the two-spotted spider mites. By obtaining clean plants from the nurseries, you can prevent two-spotted spider mite infestations. Also, refer to the Vegetable Production Handbook for a list of registered products. A study conducted by my student examined the efficacy of ultraviolet C radiation in suppressing two-spotted spider mite egg hatchability. UVC is applied at night at 2 doses, 200 and 350 joules per meter square in both Radiance and Brilliance strawberry cultivars. Results indicate a significant suppression of egg hatchability from UVC application of both doses in Brilliance and 350 joules per meter square in Radiance. Therefore, UVC could play an important role in two-spotted spider mite management in future. Cyclamen mite infestations are more difficult to control once established in the field. Therefore, the best practice is to obtain clean plants from nurseries. If frozen transplants are suspected of having cyclamen mites, they may be dipped in hot water at 111 degrees Fahrenheit for 10 minutes. The miticide Portal is effective against cyclamen mites also. Again, Please refer to the link for a list of registered miticides for cyclamen mite control. There are several natural enemies present in and around the strawberry fields as shown in the slide here. Minute pirate bugs and big-eyed bugs are effective thrips predators. Also, lady beetles and lacewings are generalist predators that will feed on aphids, whiteflies, thrips, and worms. Therefore, make every effort to conserve these beneficial arthropods to avoid secondary pest outbreaks. This brings us to the end of this presentation. I would like to thank the Florida Strawberry Growers Association and all the collaborating growers for funding and supporting our research. I'm also grateful to our industry partners, my GCREC colleagues, and the members of my lab. Please feel free to contact me via email. Thank you and have a great day.